Well, good afternoon, everyone, and a special welcome to Hendrik Reimann, our speaker of today. So Hendrik is a research assistant professor at the Department of Kinesiology and Applied Physiology at the University of Delaware. And he started his career as a mathematician, actually. And then he worked in theoretical neuroscience and robotics. And now he is moving more into our field, I would say, and especially the kind of stuff I do. So he's researching gait, and he studies how we walk, and he has a whole host of interesting studies on this. And well, I like to keep my introduction short. So Hendrik, I'm going to give the floor directly to you so you can do all the talking.
Thank you, Stuart, for the kind introduction, and thanks for inviting me here. I'm very excited to present here. I'm going to share my screen. And I can't share my screen. I think I need some kind of permission.
I hope it's possible now.
Yep. Hope everybody can see that well. Good. So I tried a bit of a catchy title, and what does our brain do when we walk? Of course, our brain does lots of things. Before I really go into this, let me also say I was told this is more of a one-way thing, but I really like presentations to go two ways. So please, I designed this modular, so there are some things I can speak about in the end that I don't have to speak about. If you have any questions or want to make a discussion point, please feel free to interrupt me at any point. So what does our brain do when we walk? Of course, it does lots of things, and here we'll focus on one thing. Humans walk a lot, and usually they don't fall, but sometimes we do fall when we walk, and the brain has something to do with that. Just very briefly, the falling is an actual problem. I just pulled together like a bunch of infographics from the internet here, not to go into any details, but to illustrate that it is, just put one number out here. It's 34 billion in 2013 in the US healthcare costs related to falls, 11,000 falls that lead to hospitalizations each year, and lots of instructions on how to prevent falls and how to not be injured by this everyday activity. And we don't really understand why we fall yet. From a we have some implications, some indications on why humans fall, but at the end of it, it's complicated. And falling or not falling, we try to understand what happens when we don't fall. So when we walk without falling, the brain does play a role in that. What the brain does, even without us paying attention, is that it regulates our balance during walking in a sensory motor control loop. So I just briefly summarized that here. The four major points of that are that the brain gets some sensory input, and based on the sensory input, it does some control. The control then leads to motor action, a change of muscle activity, and the muscles generate force that interacts with the environment, changes how the body moves, and that again leads to changes of sensory input. And we will be focusing or trying to understand here what the brain's role is in this sensory motor control loop, which is, and we need to understand what the brain's role is in order to understand how that sometimes fails. And we'll get to that in the later part of the talk. So let me remove the funny bird in the background and draw some of these things out. Motor actions, we understand relatively well how muscle generate force, what the environment does, and how forces interact with the environment. We also understand relatively well. We have a decent idea of how sensory systems get information about the environment. We'll be focusing mostly on the visual system and the vestibular sense of balance in the inner ear. There's also the proprioceptive system that is very important, but we won't be talking so much about that today. And the brain is really the missing link in the middle that connects these two parts of the body, the sensory system on one side and motor action on the other side. And the brain's role as a controller is to link sensory input to motor output. And we will first be trying to understand, or most of my research is to try to understand how this happens. This is our main tool that we use for that. It's a very labby environment. As you can see, we have people walk on a treadmill and light them up with all kinds of gadgets to monitor how they move. And we have a virtual environment, virtual reality dome around the treadmill, so that when people walk about it, we can mess with their visual system. I like to say we troll people to think that they're falling using visual perturbations and also vestibular perturbations, and then we observe what they do. And we do that to make conclusions about what the brain does, what the role of the brain is to link this changed sensory input to the motor output that we observe. So just going through this over our setup real quick, we have a virtual environment that we project on this dome. We usually use this spacey scene, what we call the cube farm, of cubes randomly floating in space. We have a self-paced treadmill so that once you walk up there, you can just freely walk and speed up and slow down without this additional cognitive task of having to match the speed of the treadmill. And the visual flow of the scene is matched to the treadmill speed, and also your perspective in the virtual world is matched to your head position. So when you move your head around in space, your perspective changes, which is important for the immersion in the virtual world. And we have some adaptations. At first, when you start walking there, it feels a bit weird, but once you've walked for a few minutes, it feels very natural. So what do we do with that? As I said, we kind of troll people. This is an example. People walk in a virtual world. And then at some point, to give them the visual impression that they're falling, we rotate the world around them, just as a human would rotate in the world around the ankle when they fall sideways. And you see that it's the rotation is not huge, ends up to just about 11 degrees. But you can see from this example that it's effective. People do follow it with their posture, and it does threaten balance. And we do this, we rotate the scene for about one step, 600 milliseconds, with constant acceleration, leading to about 10 degrees, a bit over 10 degrees of rotation. Then we stop it and observe for another 3 steps before you reset it. And the first thing we look at is what the whole body does, like the overall response of the whole body. So we did focus here on the center of mass. So this is an illustration of what the center of mass of the body does. The gray is 2 strides of walking. Center of mass oscillates back and sideways. Without any perturbations in the gray, and then we do add the perturbation. This is the orange. What happens is that this person thought they were falling to the right by the visual perturbation, since they were not actually falling to the right. So then they react to that by trying to push themselves back upright to the left. And since they were not actually falling to the right, that results in them then swaying to the left and shifting to the left gradually. And this is somewhat made to look bigger on overall for this 10 degree of rotation. What we see is that the subjects shift their center of mass by about 4 to 5 centimeters. And this is just an example from one healthy young person. And we did this in 20 more healthy young persons with shifts to either side. And I'll walk you slowly through this graph because you'll be seeing a lot of this kind of graph. So what you see here on the x-axis is normalized time, and the gray and white areas indicate the double and single stance phases. So the start of the perturbation is here at 0. and then the scene rotates for about 1 step, and then there's 3 more steps where the scene just remains the same. And on the y-axis here is the center of mass. And this is the average of 20 healthy young people. And the 2 colors are when you think you're falling to the left or to the right. And let's just stay with blue for now. Blue is you think you are falling to the left. And then you react by pushing yourself to the right, trying to push yourself back upright. And what we see is that over the course of 3 to 4 steps, the center of mass shifts by about 4 centimeters to the right. So this is the overall balance response, which is pretty much what's to be expected. We take this to think that our stimulus works. It actually was quite a bit of work to get it to be very believable using the virtual reality setup. But we now have a control loop where we can modify the sensory input that the brain gets and then observe the motor output. And we use this to do these well-controlled modifications to give false stimuli and observe what is the immediate motor output that the brain modulates to keep the body upright. This is somewhat immediate, but you see that for a while, nothing much seems to happen for the first like 500 milliseconds. Center of mass does not move much. But then over 2 to 3 seconds, it starts deviating. But what we're really interested in is what happens in these first 500, 600 milliseconds that generates this center of mass response. Because the center of mass is what's the controlled variable here. It's what the brain wants to regulate. And we're interested in what muscles does it use for that, which characterizes the more direct sensory motor link for the neural controller. So in order to understand that, we need to briefly focus on this other side of the sensory motor control loop, which links motor action to sensory input via the environment. So motor action is a change of muscle activity, and that changes how the body moves through the environment via forces. And that is then picked up by sensory input, which is usually in the domain of biomechanics. So we need to understand how forces change the moving body. And that is somewhat complicated for a walking body. So we'll do lots of simplifications. And these simplifications were introduced in robotics in the 80s, and then Ad Hof took them up for human motor control in the mid-noughts at a similar time where people also in robotics were paying more attention to what's called dynamic walking, trying to control walking. And this is a highly simplified version of a human body. Human body, of course, has many, many moving parts, but we'll simplify it for now. We'll start simple to just one point of mass that is moving on a rigid rod, which is a leg. And when you do look at the equations for that, the equations of motion, you can simplify that quite a bit to understand how forces interact with the center of mass or with the mass of the body. And what we get is this equation. And I'll just briefly go through this. Well, before going through this, let me talk about this conceptually. So, because conceptually it's actually relatively clear. How do forces interact with the body? Well, if I want my center of mass to move to the left, I need to apply a force to the ground towards the right. So I need to push myself towards the left by applying force to the ground. Another question becomes how, or that is expressed in the equation, where here the x2 dot is the acceleration of the center of mass, and the x is the position of the center of mass, and p is the center of pressure. And we see that in the simplified version of the body, just a single link inverted pendulum, the center of mass acceleration is proportional to the difference between the center of mass and the center of pressure. So if I want to accelerate my center of mass to the left, what I need to do is move my center of pressure to the right. And for instance, if I push against the wall to the right, that does generate force on the right, and that moves my center of pressure. So usually we cannot do this. Usually we do this with our feet. So now we have the simplified biomechanics. We can again look back at motor control and ask, in a situation like this, where we are almost falling, and in this specific case, it's probably already too late, but we are in situations vaguely like this all the time, where the body is falling and we need to regulate it a little bit to catch the center of mass and keep it upright. So to change the center of mass movement, we have to shift the center of pressure in the direction of the fall. So, and if my center of mass is moving too much to the right, I want to accelerate it to the left. And what I need to do is shift my center of pressure to the right. And how can we do that in a complicated body like ours? One version is to control our foot placement. This is video from a robot that only can control its foot placement. It's Cassie at the University of Michigan. And what you see is that it steps all the time and it maintains balance by slightly modulating where it steps. It doesn't have an ankle, it doesn't have hands to keep itself upright, just changes where it steps and can navigate and maintain balance, even in a somewhat problematic environment where the ground is somewhat tricky. So foot placement is already enough most of the time to keep our body upright. And that, again, that was shown theoretically in the mid-80s. And then in the mid of the noughts, people started looking at how humans do that and started showing that probably humans do use foot placement to maintain upright while walking. So what we are looking at here, this shows a foot like right before it's being placed. And in order to change my COP to the right, what I can do is just, instead of placing my foot right here, I can place my foot a bit to the right. And that will change the center of pressure throughout the next step to the right, because the center of pressure is somewhere within the foot. So if I move my foot to the right, that also changes the center of pressure. And if humans, so assuming humans do that, then what we expect is that they change their center foot placement relative to their center of mass. And this was hypothesized and then tested by Manoj Srinivasan in his lab in 2014. And what you see here is, this is not their data, but this is one example from our own lab. When you look at the center of mass at mid-stance and look at changes and then correlate that with changes in foot placement in the following step, then you have a very good correlation with about 80% of variance explained. So that when your center of mass is a bit more to the right, that means it's kind of error. So if your center of mass is more to the right, you then want to keep balance by also changing the center of pressure to the right. And you do that by moving your foot placement more to the right.
What is zero in this graph?
The zero is just the average foot placement here.
Over an entire trial or from the previous step?
This is for, the zero is the average of across all the steps here. So what this does is take a long walking trial of a few dozens or hundreds of steps and look at deltas from the mean. So changes in foot placement relative to the mean correlated with changes of center of mass at mid-stance relative to the mean. So this is good evidence that humans do use foot placement, but it's not really conclusive evidence because, well, if your center of mass is more to the right at mid-stance, then your foot placement kind of has to be more to the right or is expected to be more to the right just because your foot is connected to your center of mass. So to actually look at whether to make that final conclusion to what the brain does, what we did is look at foot placement in response to our visual false stimuli. And again, in the top here, I showed the center of mass, just stretched out a little over the 4 steps. And to remind you what we're interested here is what happens in these first 500-600 milliseconds of the first step. So the first double stance, single stance, and then double stance. What you see here is the medial lateral position of the swing foot. And you see that people do change their foot placement actively. If they think they're falling to the left, their foot placement changes to the left. And if they think they're falling to the right, their foot placement changes to the right. And this is more conclusive evidence because this is just a sensory perturbation. So what we did here is we changed the sensory input to the neurocontroller and observed the motor output. And we see that false stimulus leads to foot placement change. And we usually look at just the average foot placement within across many repetitions with the subject. And this again here is over 20 subjects. You see that it's not a big change. This is less than a centimeter, but it's a very, so let me take half a step back to put these numbers in perspective. So this is all relatively small. Like the false stimuli were not big, and overall they lead to just a few centimeters of center of mass shift. So the motor actions or the control actions that lead to these are also relatively small. And as you see here, is only foot placement changes of about half a centimeter, so a few millimeters. So you can ask, like, this is so small, what does it really matter? But it's very systematic, and it does show that the brain uses these and uses these mechanisms of neural control and uses these systematically all the time while we're walking. Foot placement is not all, because the foot placement is good and all, but we only step every once in a while, like every 500, 600, 700 milliseconds in normal walking. And as you saw from the previous video, a robot that only has foot placement and doesn't have anything, any ankles or anything, walks kind of a little funnily. So there's more. Here's another robot that gets mistreated. It's not in walking, but in single stance. And this robot is also able to maintain balance just without changing the foot placement. And what it does is a combination of muscle or torques along the upper body and the leg. And here we'll mostly be talking of torques at the ankle joint and what we call the ankle roll mechanism. This again was first pointed out by Hof in 2007 in a paper where he looked at the center of pressure within the foot, and he noticed that in healthy human people, so these are these graphs on the right, these are healthy people, and he highlighted, so the bluish area are all the center of pressure trajectories over many, many steps of walking. And then he highlighted 2, which are the solid and the dashed black line. He saw that if you start at the very positive end with your center of pressure relative to your foot, then you kind of cross over to the negative. And if you start at the very negative, then you kind of cross over to the positive. And he took that as an indicator that the center of pressure is actively modulated for balance during walking, because it kind of makes up for errors or inaccuracies in foot placement. And further evidence for that was that this crossover is missing in amputees. This is data from an amputee, transtibial amputee, who has one typical leg and one prosthetic leg with no ankle joint. And the typical leg, that the center of pressure does the same. But in the prosthetic leg without the ankle joint, you don't have this kind of crossover. The center of pressure still looks similar on average, but it's not actively modulated. Again, this indicates that humans probably do this, and there's some good evidence that they do it actively. But we were interested in, does the brain actually do that as part of a sensory motor control loop? And here are 3 outcome variables that are all kind of related to the foot placement that all tell a similar story. So let me walk you through this one by one. And this is just the first step on the x-axis here. And what you have here is the ankle inversion, eversion angle of the stance leg. And you see that if you think you're falling to the right, then you do move into more ankle eversion on the right stance leg. So your right foot is on the ground, and then you invert when you think you're falling to the right to push yourself to the left. The center of pressure is also moving, and as I said, center of pressure is moving to the right to put yourself to the left. And this also even shows up in the electromyography data. This is data from the peroneus longus down here. And in order to invert, the peroneus longus is an everter. So if you want your foot to invert, you have to reduce activity in that, and that is exactly what happens. So this is evidence that humans do systematically use this ankle roll mechanism for balance control. Okay. A brief pause to check time. How much time do I left? I think we started a little late.
Yeah, about half an hour.
Okay, sounds good. Drink and leave a second for any questions at this point.
Can I ask a question here? I have seen a lot of in the literature that they reported this as an ankle strategy, but what I saw, especially in the robot video that you showed to us, I feel like this is like can be roll off whole of the body, and it's not really fair to mention this as an ankle strategy because the robots use whole of the upper body and hip also to shift the COP. I know that this is COP shift, but it's not really, for me, it's not really fair to mention this as an ankle strategy. What do you?
Yeah, that's a fair point. This term of ankle mechanism or ankle strategy has a bit of historical roots, mostly from standing, where there's some people talked about ankle strategy and hip strategy for balance control. And then researchers did show that it's not really 2 different strategies, but it's a continuum later. And as you correctly say, it's definitely more, all muscles are involved along the whole body. I call it ankle strategy because so biomechanically, you can separate a hip and an ankle strategy. And one thing you can say is that the ankle strategy does move the center of pressure, and the hip strategy uses more of a shear force, which does not necessarily change the center of pressure. And that is one reason. And another reason is that we hear what I showed you is good evidence that is for this kind of motor response that is all centered around the ankle joint. We have the peroneus longus, which is acts on the ankle, and we have an ankle joint angle. And we have seen these systematic changes at the ankle in response to these small balance false stimuli, but we have not seen similar systematic changes at other joints related to this mechanism or similar mechanism. That's why I stick to this name of ankle mechanism or ankle roll mechanism, although it's not only the ankle. I agree with that.
Thank you.
So we looked at the overall body response to these small false stimuli, and we looked at these 2 distinct mechanisms of foot placement and ankle roll that do seem to generate. And then our next question or few questions was how both of these are used by the brain but how does the brain kind of mix and match and coordinate between these two mechanisms? So I'll go briefly over a few experiments that we ran to tease out this question, or to answer this question from different points of view. Just briefly, I'm showing you another modality to induce these false stimuli that we use, that some of you might be familiar with, which is galvanic vestibular stimulation. This is an oldish video from a group that demonstrates it as human remote control. What happens is that you have two electrodes behind the mastoid processes. You can see this person wearing them. And there's a light electric current that runs mostly through the vestibular nerve, and that changes the vestibular signal, which gets interpreted as a feeling of falling sideways. And the direction of falling depends on the polarity of the stimulus. And we do that to induce these small false stimuli at distinct points of the gait cycle, similar to what I showed you earlier in vision. And again, we leave them on for one step, or we leave them on for about 600 milliseconds, which is a bit less than one step. And here we were asking, how does the coordination between these two balance mechanisms change depending on when in the gait cycle we stimulate? So we have, again, 20 healthy young subjects, and we provide this galvanic vestibular false stimulus at heel strike, or with a delay of 150 or 450 milliseconds after heel strike. And the idea was, one of the ideas was that 450 milliseconds is about 200 milliseconds before the next foot placement, and step timing was controlled by a metronome here. So the idea was that it's just too late to still change your foot placement. And we were trying to understand if the brain, and that means the next foot placement would be in about like a bit less than a second, which is quite a long time. So if we remove the foot placement or change the timing until the next foot placement, how does the brain coordinate between foot placement and ankle roll? This is a kind of busy slide, so I won't walk you through every detail, but on the top here, you see these graphs, or most of the graphs are similar to what you've already seen before. On the top here is the whole body response. Now we look at the first two steps, and what you see here is the velocity of the center of mass. And the three different color group, the two different color groups are false stimulus in two different directions, left or right, and then three different shades are the different time points, early, mid, and late. Here's where they happened, early, mid, and late, relative to the heel strike. And the two directions are blue and reddish. You see that the center of mass overall changes similarly for early and mid. So 150 milliseconds later does have some effect, but not a huge one. But something does seem to be different. There's a smaller later response, but it's also smaller for the late response. And we look at that. We look at the ankle roll, which is here represented by the center of pressure shift. And again, we see that about 250, 300 milliseconds after the stimulus, you do see the center of pressure diverge to the left and to the right. And again, this is changes in center of pressure relative to the center of mass. So the false stimulus comes, you think you're falling to the right, center of pressure shifts. You think you're falling to the left, center of pressure shifts to the left about 300 milliseconds after the false stimulus. And the magnitude is similar here. So these are the trajectories, and down here we have the magnitude, which attempts to quantify the amplitude of this shift. You see that the magnitude of the overall shift is vaguely similar for the center of mass. It's smaller. Interestingly, for the center of pressure, it is larger in the late versus the early and mid. And then we look at the foot placement later on. you see that in the late condition, there's pretty much no foot placement, which is as expected. Like foot placement is on average really just zero change. It's a bit less of a foot placement change if you have a little delay. And what's interesting here is that there's no foot placement. There's a larger shift in center of pressure. So there is some shifting between the two mechanisms here from foot placement to the ankle roll. And what is not really shown here, but in this late situation, the ankle roll is really taken over by, so here the ankle roll is done by the stance leg, but in this late situation, the ankle roll is taken over by the then new stance leg. So the neurocontroller coordinates between foot placement and ankle roll, but also between ankle roll of the old and new stance leg. And all of that is where the whole body response is approximately similar. But we do see that when there's less foot placement or no foot placement change, the overall response is somewhat smaller. So there is some kind of ordering between these two mechanisms. While the previous one we looked at, or we used the same stimulation at different times of the gait cycle. In the next experiment, we were looking at it, coming at it from a slightly different angle, where we asked, how do use of these mechanisms for balance change with gait pattern? And we're mostly interested in faster versus slower walking as a kind of a long-standing debate and what is more stable. Are you more stable when you walk faster or slower? And here we modified that by controlling cadence. So we use a metronome, and we have people walk to a metronome, and we gave them two different metronomes for a low and a high cadence at 90 beats per minute or 110 beats per minute. Actually, there's an error. It was 80 versus 110 beats per minute. And you see that people do adjust their step time to the metronome. The low, like the 80 beats per minute, is very slow. So people didn't like walking that slow. So on average, they went a bit below that. They adapted their step length and their velocity. So cadence controlled, but it's people, since it's a self-paced treadmill, people were free to walk, to vary their speed. And they did mostly vary their velocity and step length to hit this cadence. And then what we are looking at is how do use of the balance mechanisms change for these two different gait patterns. The first thing I want to point you to is the overall whole body response of, and this is now a longer window, this is for a full 6 seconds after the stimulus. So the stimulus is only here, like the first 600 milliseconds. And you see that in both cases, center of mass deviates in the direction of the perceived fall kind of substantially and then comes back. And the striking thing here really is that there's not much difference or non-significant difference between the yellow and the blue. So even though your gait pattern is substantially different, when we change the sensory input by giving you this vestibular illusion, the overall balance output is pretty much the same. The overall body sway to the side is very similar over the following 6 seconds. But we found there's a very substantial difference in how this sway is generated. So the yellow here again is the low and the blue is the high cadence. We find that here is foot placement, that in the high cadence, people used foot placement very substantially. This is an average of a 3 centimeter foot placement change in response to the vestibular stimulus. Whereas for the low cadence, there was actually no change in foot placement. And one of the reasons for that is that in the low cadence, and here, this is the ankle roll, and this is now normalized in time. So while this is one step and another double stance, the yellow and the blue have different lengths in real time because of the different controlled step times from the metronome.
Hendrik,
So,
may I ask a question?
yes.
Did you have to adjust the GVS stimulus to get the same overall response at a different speed?
We did not. So we were actually somewhat surprised that we did get the same overall response. We didn't really expect that. And we did not. We just used the same GVS signal.
Yeah, but because there are studies that show that at faster walking speeds, the GVS has less effects, right? Or maybe that's even higher walking speeds then? I mean,
there are, yeah. I'm familiar with some studies. I don't know if we're talking about the same ones. It's complicated. I'd maybe not get into that right now because that would be a longer discussion.
Yeah, sorry.
So in the low cadence, slow walking condition, you have a very substantial ankle roll response. And in the faster walking, you also have an ankle roll response, but much less so. So the same overall or very similar overall whole body response is generated mostly by ankle roll here in the low, slow walking, and mostly by foot placement in the faster walking condition. So more evidence that in these healthy young people, the brain is, or the neural control is actually very good at coordinating these different mechanisms depending on when things happen, as we saw earlier, and depending on the overall gait pattern. Just briefly, another factor. So since the title of the talk is what does the brain do when we're walking? All of these things I talked about so far is what we're not really aware of. It happens in the brain because it links like the vision and the vestibular system to motor output, and the vestibular system goes through the brain stem, but the visual information is processed in the cortex. One thing we all were also interested in is how can these things be modulated by cognitive factors? So as a very careful, small start into that, what we did is gave people these lanes. We separated, we put a virtual path into the VR system, and this now uses virtual reality headset. So we could, because our dome is very nice, but it doesn't cover the floor. And what we did here is ask people to usually walk on the green lane. If they have to, they're okay to step into the gray area, but do not step into the red area. That's all we told them. Like, we did not say anything bad would happen. We just said, do not step into the red. And then we gave them vestibular false stimuli about when they were around in the center of one of these areas. And then the false stimulus could be either towards or away from the no step zone. So towards the towards the no step zone, which is red, or towards the neutral zone, which is gray. And we looked at how does this cognitive task change the balance control. Here, similar curves to what you saw before. In the top right is the whole body response. And what we do see in gray, so again, we have 4 steps, changes in the center of mass position. And we do see that if you think you're falling towards a red no step zone, then your overall body sway in response to that is larger. It's not enormously larger. So this tops out at about, we use a stronger GVS signal here. So this tops out at a bit less than 7 centimeters. This is around 5 centimeters. So it's a significant difference, though not a huge one. We did see a significant change in foot placement. And this actually surprised us a little. And this is significant. There was also a bit of difference in the ankle roll, but this did not come out as to be significant. So let me briefly mention why the foot placement change surprised us. So what we had initially hypothesized would happen is that when you think you're falling towards the red no-step zone, that people would not use their foot placement because that requires you to step closer to the no-step zone. So if I have a red zone on my left, I think I'm falling to the left, and the foot placement mechanism says, put your next foot placement more to the left to accelerate your body to the right. So we had thought that the foot placement in the no step zone condition would be reduced because the brain would shift to the ankle roll mechanism instead. But we found the opposite. We found that the brain actually uses the foot placement more. And in hindsight, that did make kind of sense because these lanes are relatively wide, like 33 centimeters wide. So, and your foot placement change on average is just 1 or 2 centimeters. So you're not really stepping dangerously close to one of the no step zones. So in hindsight, this made sense that people do this. And it's evidence that while most of these things happen unconsciously, subconsciously in the brain, that's automatic neural control, this control is subject to cognitive change. And people do kind of tighten up a little with this very simple cognitive task of not stepping into the red zone. We briefly considered like making the red zone a hole instead, but we thought let's keep it simple and just use this, like not get into fear of falling, just use this very simple instruction. So all of what I take a brief sip and open for questions. So in the remaining time, I want to briefly talk about balance control in a system of people who are not considered typical, but have some kind of neuromotor impairment. Specifically here, I'll show you data from cerebral palsy. All the data I showed you so far was from people who are considered neurotypical in normal walking or the typical system. But how do these things change in people who are not typically developing, but have something that increases their fall rate or have problems with balance? And people with cerebral palsy is a neuromotor impairment. And some of these people fall all the time. Much higher problems with falls than me. So CP is a high rate of falls. It's associated with proprioceptive deficits. And these people tend to rely more strongly on visual information. One of the hypotheses is that because their proprioception is somewhat impaired. So what we were interested in is if we give these visual false stimuli to people with cerebral palsy, what do they do differently for balance control compared to typically developing age-matched controls? This data from 28 subjects. We have finished data collection, but I'm currently analyzing this, so this is still somewhat preliminary. Here you see 14 people with cerebral palsy in red and 14 age-matched controls with typical development in blue. And this shows the whole body response. And here we show 8 steps after the stimulus. And you see that the overall response is pretty similar at the previous center of mass plots. We used to cut off around here. So overall, the curves look similar, but compared to the age-matched controls, people with CP respond kind of more strongly and a bit delayed. And this made things a little tricky to analyze. So what we did here to quantify that is we calculated 3 variables, which are, I'll show these here, the area under the curve. So we integrated the difference in center of mass, the center of mass response over the whole 8 steps. We calculated the peak displacement of the center of mass for each person relative to the unperturbed steps. And we also calculated the timing of this peak of the maximal center of mass excursion. And we did that for each subject individually. And what you see here in the box plots are the comparisons. And without going into all the details, what you see is that we do have a significant group effect here. So, for instance, the area under the curve is significantly different for people with CP versus typically developing. So people with cerebral palsy respond more strongly to visual false stimuli, which says that something in their neural controller is different. In addition to all the anatomical changes that and the changes in the periphery that happen with cerebral palsy, they also have changes in the neural controller, mapping high-level visual input to motor output for balance. Again, we were interested in how the balance mechanisms change. Now, again, this was tricky because we usually look at the first foot placement after the perturbation. When we look at this here, we do see a bit of a difference, but it did not play out in the way we expected and in a way that made sense. So what you see here, so this is foot placement difference for the first 4 steps. And here we pulled out the foot placement difference for the first step. And on the sides here is the more affected and the less affected side for the people with cerebral palsy. So here we actually saw a larger foot placement response in the typically developing group in blue, and a smaller response in cerebral palsy, which really puzzled us because overall the people with CP had a larger center of mass response, and they had a smaller foot placement response in the first step. But there was also this delay effect that their center of mass response happened later. Then when we looked at the foot placement in the second and third steps, and we did that here to pull that out, we saw that although in the first step, CP is smaller, in the second and third step, their response is actually larger compared to the typically developing peers. And when we pulled that out, and what we did for now is just take the average or the sum of these 3 foot placement changes, is that the summed foot placement changes over the first 3 steps are larger in cerebral palsy. This is our preliminary analysis. We haven't completely figured this out. This is currently in preparation for a publication. And then looking at the ankle roll as well, this was intriguing because here you see that, again, cerebral palsy in red, typical development in blue. See that people with cerebral palsy have almost no ankle roll response, whereas typically developing age-matched controls do have a bit of an ankle roll response. One caveat here, what we did here is not use a metronome. So people here typically walked a little faster. So, and as you saw previously, for faster walking, people use less ankle roll anyways. So we expected less ankle roll overall, which is what you do see in the typically developing group. And in the CP group, we don't see any ankle roll at all.
Hendrik, may I ask a question?
Yes, please.
I'm surprised with this graph and also several of the previous that the typically developing seem to have larger error bars as a group than the patients. And normally you have severe and less severe patients, so you expect to have large variability in the patient groups. And even in one of the previous one, you saw that the affected and non-affected side of the non-affected, the typically developing participants showed a larger difference than that of the patients. So it's,
yes,
it almost looks like the colors are switched, but
yeah, we are aware of that. That has also puzzled us. I can assure you the colors are not switched. We've looked at that several times. but I don't have an answer for you. It is very puzzling and is something that we're looking into currently, but we do not have a good explanation for this at this point.
But people with cerebral palsy quite often also have like quite spastic calf muscles, right? So doing
Yes,
inversion eversion for them is basically anatomically already quite a hard thing, or it depends probably on your specific subpopulation, but
it does, and this was a mixture of different groups. So we have some hemiplegic, some diplegic in here, different levels of their functional scale. Yes, so that leads into the question of why we do this, and or why we do see this difference, and that is one possible explanation, that there's a difference in the overall rigidity at the ankle, and that. So I'm looking a bit at time here because that leads into something that I wanted to show you later, but we're pretty much out of time. So I had some modeling results, which I'll probably not go into anymore, unfortunately. But let me briefly jump ahead and show you this other results from cerebral palsy, because that is one possible explanation for these differences that Sjurt just brought up, of this increased rigidity from spasticity. As I mentioned earlier, people with cerebral palsy also have impaired proprioception, and we hypothesized that maybe this impaired proprioception is more important for the ankle roll mechanism, and reduced proprioception prompts them to rely more strongly on visual information. So one thing we tried was to improve their proprioception using electrostochastic resonance stimulation. That is, what we do is we apply some electrodes along the shank, on the calf muscles and the ankle joint, and we run a stochastic white noise electrical current through that. And that has been shown to improve proprioception, to improve the function of muscle spindles, and it can boost balance control and standing, as has been shown previously. So this is, yeah, let me not go into that. So we did that in people with cerebral palsy in walking, and then gave them our visual force stimuli. And what you see here is what the stochastic resonance stimulation at their calves and ankles did to their response to the visual stimulus. First thing I'll point out is in the CP group, the overall response to the stimulus went down somewhat substantially and significantly. So this is from 11, this is again preliminary data from 11 subjects, and you had a somewhat stronger response here in the people with CP without stochastic resonance, and then with stochastic resonance that went down significantly. And this is a reduction actually for each individual. You see the individual subjects here linked. So each and every subject had a reduction here, which we found quite surprising and exhilarating. And in contrast, in the control group, there was no effect whatsoever. And this is what we kind of expected. And our rationale for that is that in the control group, proprioception is already good enough so that boosting it further does not add anything anymore to the balance control overall. All right, I think I'm pretty much out of time, even having started a little late. I had some modeling results, and then just briefly add that this is actually something that we had predicted with a model a few years before, that when you don't use your ankle roll, then your foot placement is a little larger. So it's something I also do, which is a lot of fun, the modeling work, but going into that now, we don't have time to go into that now. Well, thank you for the questions that were already there, and I'd be open. We're already over time, but I'm happy to stay here and talk.
Hendrik, thank you very much for your talk. I guess there's still people around with questions. Yeah, the floor is open for questions. I see a lot of hands, but they're not raised, they're clapping, which is good. Yeah.
Okay, thanks, Hendrik. In your last part, you almost seem to suggest that maybe the control of the ankle roll is, the ankle mechanism is separate from, so it's guided by proprioception, whereas the visual, the stepping responses seem to be guided by the visual and vestibular input. But in the first part, you clearly showed that they're very well coordinated. So do you think of these as two separate control mechanisms, or is it really part and parcel of one controller?
It's a great question that I would say both, actually. It's not necessarily an either or. So many of these earlier results that I showed indicate that there is what I would call an overall whole body response, and there is a different part of the controller that, so the way I see it currently is that the brain kind of generates a desired acceleration, how much it wants to accelerate the body, and then it recruits whatever mechanism is available to generate this overall response. And at sometimes, or at some, depending on the point in the gait cycle and the overall gait pattern, maybe the ankle roll is more available than the foot placement, or the foot placement is more available. And there's a different part of the controller that shifts between the two and possible other mechanisms, depending on what's available.
But doesn't that make it strange that the, let's say, the sensory information seems to be the factor determining this difference in use between CP and healthy?
So I didn't catch, could you repeat that?
So
I didn't catch that.
Isn't that at odds with the fact that the difference between the CP individuals and the healthy individuals seems to be determined by the use of sensory information, maybe rather than anything else? I mean,
yes, so that's a good point. I would say there's yet another part that is before that. So what I said is that the brain or the neurocontroller kind of generates a desired acceleration of the center of mass. But where does this desired acceleration come from? It comes from an estimation of the current movement of the center of mass. So you use your sensory systems to estimate, well, currently I'm moving yay much to the right. That's a bit too much. So I want to accelerate my mass to the left. And that gives this desired center of mass acceleration. But how do you arrive at this estimate of your current center of mass kinematic state? You use your different available sensory systems, so the visual, vestibular, and proprioceptive system. And these different sensory streams have to be fused in some way. And what we believe where the CP effect, or the effect that we saw in CP comes in, is that this impaired proprioception changes how much people rely on one system over the other. Usually proprioception is relatively good, and vision as well. But so there's one paper from like the late 80s from Fitzpatrick who tried to look at that in standing. How well can you use your different system to detect that you're swaying and standing? It showed that vision and proprioception were approximately on par in standing. Vestibular was a lot worse. So that would mean that usually in standing or walking, you rely on vision and proprioception to about the same degree. But now if your proprioception is impaired from this neuromotor problem, your proprioception is noisier or worse. So then you rely more on vision. If you rely more on vision, and then we mess with vision using the virtual reality, then you rely more on an erroneous signal. That means that they respond more to the visual perturbation, as we saw. And you could say, well, what does it matter? Well, vision is kind of slower and more complicated, and you use it for lots of other things too. You use it to navigate, to look at your phone, to do all kinds of things, to look for things to hold on to. So vision is busy, and our overarching hypothesis there is that people with CP have to rely more on vision, and that leads to problems with balance because their proprioception is worse. So if we can improve proprioception, we might be able to allow them to rely less on vision, free up vision for other things, which reduces their liability to fall.
Okay, thank you.
Other questions? I do have one. So I was wondering the proprioceptive aid with stochastic resonance. Do you think that it would also work for elderly? I mean, they're set to rely more on vision as well, right? So
yes,
assume that their proprioception is less, I guess.
Possible. I don't have an answer. I think there's a good chance. We are currently collecting data from people with Parkinson's disease who also have some problems with proprioception. We just have 3 subjects, so I wasn't comfortable showing those data, and currently it looks a little funny, so I'd rather not talk too much about that. We have not looked at proprioception in older adults. We are currently running the preliminary study for that, which is to see how these responses change in older adults to begin with. It's been very challenging to recruit older adults over the past 2 years, which is why that study is not as far advanced as I would like it to be. Stochastic resonance might be the next step there, but we don't know yet.
Okay, thanks. Marcia.
Hey, thank you very much for your nice presentation. I have a question about one of your slides before cadence when you were presenting. It was about the left and right movement.
Oh, yeah. Which slide was it? Left and right?
It was before the cadence slide.
This one, I believe?
Yes. I was wondering, this left and right side in all the figures are quite different. Do you know the reason, or is it?
So one reason here is that, and I didn't mention that detail, so thanks for picking up on that, is that here, so in all the other data I showed you, we randomized the triggering foot between the left and the right. So you could, like, the stimulation was triggered on a heel strike, and then it was randomized which heel strike it was, left or right. And this direction of the stimulus was also left and right. And then we analyzed whether that actually plays a role. And we figured that left or right, like we were looking at handedness or footedness for balance, and we found that it actually doesn't play a role. But here in this data, we only triggered on one side, but then still had a stimulation in the left or the right direction. So differences you see here are mostly due, or what we hypothesize is due to body symmetry. That you trigger on your right foot, and then stimulation can be towards your right side or towards the left side. And that is a difference in stability. If you think you're falling towards your stance foot, that is not being perceived as destabilizing as when you think you're falling away from your stance foot. So if I would rerun the same study again, I would probably organize that a little different, but back then we weren't that far yet.
Yeah, all right. Yeah, thank you.
More questions? Yep. Do you have a question again, or your hand is
Yeah,
still?
I do have a new point, maybe a comment or a question. I'm just wondering, the foot sole sensation that you're basically talking about when you mention proprioception, should we be calling that proprioception? Because I think it's actually tactile information. It's an exteroception.
Yes, so when we talk about proprioception, we do mean proprioception. Because so specifically for the stochastic resonance, because we put these electrodes on the calf muscles and around the ankle joint, and they stimulate the muscle spindles and the joint receptors.
Okay, I'm sorry. I thought you were stimulating the foot soles.
No, yeah, I briefly mentioned that. So when I talked about proprioception later, it then blended into it a little, because I think for the ankle roll, what is important is also perception of your current center of pressure. And for that, I think the foot sole receptors are critical. But I can't say much about teasing these apart.
Okay, thanks.
Okay, any more questions? I don't see any hands raised. So I'm gonna thank you, Hendrik. And yeah, I'm looking forward to seeing these results on the Parkinson's also at the ISPGR, I hope, right?
Yes, yeah.